Welcome back to Cyber Prophets. This time, we are going to go to a new topic. But as you know, this is a podcast made for you and only for you that is about prophesizing the future of technology. This time, we're going to speak about biocomputing. With me is Edgar Regalado, who is our co-host for this time.
Hello, everyone, and welcome to the season 2 of Cyber Prophets. We are very excited to come up with every time better and more interesting topics. And as Federico mentioned, we're going to talk today about biocomputing, which is something not many people know about. So we are very glad to have an expert among us. Federico, can you tell us a little bit more about our expert?
Our expert is a very, very motivated person, the Dr. Tony McCaffrey. He's a computer scientist in the United States with 2 degrees, one in computer science and one in cognitive psychology. I met him when he was speaking about an interesting topic about brainstorming. He created many things, and this is one of those topics that I was very, let's say, excited to know more. But this time, since our project is about technology, we are going to go into a more important topic that is biocomputing. But maybe the doctor can give us an introduction about himself and tell us more about him.
Yes. So, as Federico said, I went between computer science and cognitive psychology. I was first in computer science and in artificial intelligence. And I had some big questions about artificial intelligence and where it was going. So I decided to switch fields and study intelligence and problem solving from the human perspective, cognitive psychology. And then one of the things I did was come back and integrate them together. And I have developed this online platform in which AI programs and humans and machine learning program can work all together and help overcome each other's weaknesses. So after I got that kind of figured out and that project behind me, then I started looking for the next big thing, the mysterious future of computing, and that's when I started thinking hard about biocomputing.
So you think that biocomputer might be the next big thing?
Yeah, it's right now it's the most quantum computing is what everybody thinks about these days as the future of computing. And so I first investigated that, and I found that I did a little experiment. And so I was going to publish a paper on quantum computing and something else I was working on. And I needed some assistance understanding one of the algorithms, quantum algorithms. Mm-hmm. And so I went on LinkedIn and I contacted 70 people who had quantum programmer, quantum physicist in their profile description, and none of them could help me.
Oh, wow.
So help explain it. I think there's some people out there who can. So my conclusion was maybe it's so unintuitive that there's just a few geniuses, gurus who can actually make these programs, and everybody else kind of tweaks them and implements them and makes small modifications. Mm-hmm. So I came upon biocomputing and I said, this is less known, but it may in the long run have the biggest potential for the future of computing.
Yes. Well, yeah, this is quite a very new topic, and I think that most of us are not aware of what it actually means. So, Dr. Tony, could you please explain us what is biocomputing?
Okay, yeah, so I'm going to talk about two facets of it. One is which storing using DNA to store data. And the other fast, that's somewhat known, but there's a big future in that, I believe. And then what's hardly known at all is using living cells as the hardware or biological cells or wetware performing computation. And can you do useful computational work?
Wow.
Like the traveling salesman problem or the shortest distance between two locations or factoring? Can you use what we humans would say useful computational work with the little machines that are working inside the cell? So if it's okay, I'd like to talk about the DNA storage aspect first.
Yeah.
And then get on to the other one that's less known. So initially, I just amazed. This is also gives me a lot of awe and wonder, is that what has nature given us? It's given us the code of life, okay? These 4 bases that put in different combinations make enzymes and then proteins, and they make us. But I think, and a lot of people agree, that nature has given us so much more. This little DNA strand within us could be the future way that we record all our data. Because here's some facts. So I watched an interesting TED Talk, and the person giving the TED Talk, Dina Zelensky, she held a little vial between her 2 fingertips, so it was very small. And she said all the movies ever produced could easily be encoded in the DNA in this little vial that fit between her fingertips.
Oh.
And that just blew me away, because I didn't do the calculation for how many gigabytes or whatever that would be, but that's an incredible amount.
It's a lot.
And basically, yeah, when you convert to binary, you could store a few gigabytes in one cell in the DNA strand, and the human body has 30 trillion cells.
Trillion.
And so some calculations, these are such big numbers. 1 gram of DNA could store 215 million gigabytes.
Okay.
Not 1 gigabyte, 215 million gigabytes.
Wow.
And the DNA storage lasts a long time. The best estimates that it's could last a couple of 100,000 years before it deteriorates. So currently we can store data, but we keep updating it. So the reader, floppy disks, I can't read a floppy disk anymore. I don't know anybody who has a floppy disk reader.
Yeah.
And CDs I guess I can still read a CD, but we keep updating our technology, so the data might be there, but we're losing the machines to read that data are getting out of step.
Exactly.
So the one reason I'm really excited about DNA storage is that currently server farms consume so much energy, and they produce so much heat. And it's amazing to compare data being stored on a server with the energy consumption and the heat production versus the energy needed to keep this little cell alive, and inside it is a couple gigabytes of data. I mean, it's so that's one reason environmentally I think it's an amazing future for DNA storage. Now, currently, DNA storage could only be used really for archival, like long-term storage, where you don't have to access it very often.
Yes.
Because when our reading capability of the DNA is pretty slow at this time. But of course, I think that will get faster and faster as we go forward. So.
And how do you think we can create this, let's say, artificial DNA? Because this is something that I'm curious to know.
Right. Right. So we have the ability to read DNA, to write it, as in to put the bases together and create a DNA strand artificially. And we also have the ability to edit DNA. And so we can do everything we need to. And the technology for it, the acronym, don't ask me I forget what it stands for, CRISPR. Okay. And basically they found a way to kind of trick a bacteria to go in at the very precise location of the DNA strand by matching a pattern and cut the DNA. So, and then you can add more segments of it, so you can edit it at different locations inside the strand. Now, the people who invented CRISPR technology just, recently won a Nobel Prize for their work, and it's got so many applications in medicine, in helping people improve their health, in creating vaccines, in all kinds of things. But I'm more interested in, because I'm a computer scientist, that are kind of the computer science implications of doing this. So I don't know what it would look like. Will you have some kind of little thing on your laptop inside that has a little housing where it holds some living cells, bacteria cells, and make sure they have the right environment and enough food to survive, and then and then you store your DNA if it's attached to your laptop. I don't know what it's going to look like. Or is you going to have a little bacteria farm on your desk? I can't envision that right now, what it's actually going to look like. But
and Dr. Tony, I am very curious about your personal experience, because you have told us already, like, about the concepts and how you were contacting people on LinkedIn, and also about these technologies like the CRISPR that is used for dealing with the DNA. But have you been working professionally, or have you been in touch with this biocomputing stuff, let's say at an academical level? Or is it all theory at the time?
Right. So I did about 4 months of reading about, everything from getting a book on genetics for dummies.
Okay.
So to relearn my biology that I didn't think I'd ever use again, So 4 months of it was just theory, of reading as much as I could about the general biology of the cell, so I could start to understand how a DNA could be written, how it could be edited, and things like this. Now, on the next phase is I've talked to online a number, they call themselves synthetic biologists.
Synthetic,
and they are,
okay.
Yeah, so not the biologists and synthetic, and then they want to program, they use these words, they want to program the cell to do this thing or that thing. And so they are studying, okay, if this DNA sequence will trigger the cell to do amazing things, like to produce a microorganism that'll produce this vaccine. Or this DNA sequence will trigger the cell to produce a little microorganism that can turn methane, a greenhouse gas, into oxygen. So they use this compute. They say the cell computes, and they mean something different than computer scientists say, but there's a lot of similarities. They're trying to change the code of this biological machine or system so that it produces things on the output that are useful to them, are helpful for us. So my next step after that, after conversing with this, is I would like to move into this industry and contribute to it more if they like. I'm thinking of writing some grants or applying to some of these companies that are combining computer science and biology together.
It's a very interesting, like what you were saying. I'm wondering, do you think that we are going to be able to use this technology to, let's say, to make, how to say it, to terraform Mars in a way? Because right now the target is trying to terraform that planet, but we have only CO2 and we need oxygen to live. And from my understanding, because I wanted to be an astronomer, the easiest way will be to collide several asteroids from the belt to try to make some water or to try to produce something.
Yeah, I watched a very interesting TED Talk by Dan Gibson, who has the company Synthetic Genomics. So synthetic making, and has to do with the genome, the genetic material. And his company is very on the forefront, and they have just recently come out with what's called a digital to biological converter. And they can send their machine a file, and it has all the bases in the jars, the DNA bases, and it writes and connects the bases in a particular order, so it becomes a DNA strand. And then their idea, well, this is real, they can do this, that they can produce a DNA strand and create a bioorganism that would produce oxygen or produce medicine or produce food. Now the new thing is that for future pandemics, Dan's idea, which is going to be real, it's real now, it can happen. They made this printing, like a 3D printing machine.
Yes.
They call it a DBC, digital to biological converter. And in their lab, they might have the DNA sequence needed to produce an organism that produces a vaccine. And they'll send that digital file across the world to where the outbreak is happening. And if they have I'll call it a bio 3D printer over there, they can start producing the vaccine on site. Okay? So that's real. All right? Then we move to the next step, which could happen, but it's a little more science fiction at the moment, where we send these DBCs, these 3D bioprinters, we send them ahead of us humans to Mars, okay?
Yeah.
And they start using the resources of Mars, and then we send a digital file. Oh, we're going to take this thing that's in the atmosphere of Mars, and we're going to convert it to pure oxygen, and you make the supplies or the food or the medicine, or even you're talking about building materials, biologically printed building materials. Wow. And so when humans arrive, they don't have to carry all this stuff with them. it's made on the planet. Now, the next science fiction step is what Federico said, is if you sent enough of these printing devices, I guess you could start to terraform the planet and convert the atmosphere itself.
Wow.
But he's talking about, producing the oxygen in tanks so that we would have resources. But the next, logical step would be to terraform, I think. So it's amazing where we're at, that we can send these printers are fairly small, and they can be sent to places that need to produce a medicine or a vaccine quickly. But we're just by extending your imagination, all of a sudden we're moving toward terraforming a planet. It's amazing.
Wow. Yeah. And Dr. Tony, is this digital biological converter, the 3D printer, is something real already,
Yeah.
or? Yes.
Yes, it's real.
Incredible.
So I think I haven't heard of it's being used during this pandemic. You hear about all the vaccines are being, shipped on huge planes flying here and there. But God help us, the next pandemic, I think these machines are real, and they will come into wide use during the next global situation. And we'll be sending files around, and they'll be making the vaccine on site.
Yeah, that it sounds very useful.
Yeah, it's incredibly useful and visionary on his part.
Yeah, it sounds something like very unique. And let's say, I got like another kind of science fiction question. That is, do you think that we will be able, using biocomputing, to clone dinosaurs like in Jurassic Park? And why I'm asking this, because what happens is, from my knowledge, that you have the DNA, and the DNA deteriorates after I think 5 million years. So even if we take let's say the insect or the mosquito, what it has inside is useless to duplicate any being. Do you think we will be able to, with biocomputing, to reorganize properly and to clone, not necessarily dinosaurs? I don't think anyone would like to have a T-Rex in their grave.
If the DNA is has not deteriorated, then it might be possible. Then it could be possible. To recreate the creature. The oldest DNA recovered so far is, I have it on here, is a mammoth that was frozen in Siberia, and they were able to recover DNA that was 1.2 million years old. Now, I don't think the DNA is in good enough condition, or maybe parts of it are, but not enough of it to actually recreate that animal. But these are questions that are not, they used to be complete science fiction, but now is it science fiction anymore? And you go, I guess it still is for particular reasons, but we're getting closer and closer,
Yes, all of this sounds very surreal. But have you watched the movie I, Robot, Dr. Tony?
Oh, yes. The one with Will Smith?
Yes, that one. Do you think that with following the same line with the science fiction, do you think that biocomputers can help us build like robots, such as the ones that we see in that movie?
So in that movie, was the robot part metal and then part biology? Was it a combination? I don't remember the details of the movie.
I'm not sure also, but I think it was like that it had parts, biological parts as well, and like it had the ability to actually learn by itself, just as like something like machine learning.
So there are all kinds. So this opens up a big, all kinds of ethical questions. And so after there's been the people who won the Nobel Prize for CRISPR, they started a movement where all biological scientists cannot try these things, a moratorium where they say, no, we're going to pause and we're not going to try these things until we think through all the ethical issues. So that said, they're trying to be very, very careful. But that said, Edgar, yes, we have the potential to edit the human genome so that we could try to make parts, organs, or the brain to try to make it smarter, better memory, better at pattern recognition. We have the potential to start making edits and trying them out, which nature would call a mutation, to try to create these things, create and improve our abilities. It's exciting, and it's also very scary.
Yes.
So I'm glad that the Nobel Prize winners have put a stoppage, and the scientific community agrees until we think through the ethical issues. Nobody in the scientific community, they will go there, supposedly. Yeah, supposedly.
Yeah, the thing is that, well, like, let's say that supposedly, I will say it will depends on the scientists that are the people that are maybe the people who are more known, but not the ones that are in the background. But thinking in something else, I have a question. What are your thoughts about quantum computing, and why do you think we put so much effort on this area when biocomputing seems more, let's say, more important? It seems like something that we can really create amazing things like the digital biological converter and, well, a lot of incredible stuff.
Well, let's see. Quantum computing, the first quantum theories came out, we'll say, in the 1920s. And so the notion, and it's so strange how they work. So to take, computers work with bits, and then a qubit, a quantum bit, a bit can be in two states in a normal computer, and a quantum bit can be in those states simultaneously. It can be both 1 and 0 simultaneously. And then if you have a longer thing, say 8 bits, it can be in 2 to the 8th states simultaneously. So people, by our normal way of thinking, they said, oh my gosh, it was originally thought that every algorithm could be sped up with a quantum computer. So people got excited pretty early after they realized this. And quantum, I asked my friends who are not scientists, everybody knows something about quantum mechanics. Oh, that's that weird stuff where it doesn't act like our normal size world, right?
Yes.
And biocomputing it hasn't gotten the press yet. I think it will. It doesn't got the publicity yet. True. And I guess that's one thing I'd hope scientific journalists and myself, maybe I can help make it a little more known, the potential. And let's see, I don't remember the year that the DNA strand was first the shape of it was first figured out by Crick and Watson. I forget the year of that. I think it was later than the 1920s, 40s or 50s, maybe 60s. I don't know, somewhere in the 40s and 50s. I could be wrong. We'll have to fact check me, right?
Mm-hmm.
And then so people knew, okay, that's the core of who we are. We got this coding inside us that gets replicated and the instructions are followed and it builds things. But it didn't click that we were a long ways from the beginning of being able to program it, to control it in some way, to edit it. And that's only happened in recently. CRISPR, oh my gosh, when did that happen? 2017.
Oh,
So
yeah.
that was 4 years ago.
4 years, yes.
And so 4 years ago that we could edit the actual DNA strand accurately. Not too much before that, reading it, we just, within this century, at the beginning of this century, we finally mapped out the human genome for the first time. So we were able to read it completely, all 2 gigabytes of information. So it's kind of, it's all the discoveries we needed to move forward at biocomputing have just happened in the last 15 years, 20 years, So it's not as well known by the public.
Yes. It's a very recent field of study still. And as you mentioned, like it doesn't have the coverage of the media yet. So
right. And I think a lot of people are really scared of the ethical questions.
Yes.
I mean, can you choose your baby's eye color and go in and edit their eye color? And if they have a condition, do you, such as Down syndrome, do you go in and edit that and get rid of it? Or do you let the person Down syndrome be born and have a life with that condition?
Yes.
And so there's lots of ethical questions. And then the next ethical question is, if a government wanted to only have people with a certain skin tone or a certain hair color, and they only allow people, they have, it could be, there's lots of really, and people are scared of that, and rightly so. And so we have to really think about that carefully and not let people with bad ideas take over this technology. So there's a lot there, but I recently have thought that it is the benefits could so outweigh these ethical issues if we keep them under control. That I think it's really exciting. I envision it, since I'm a science fiction writer.
Yes.
We get this alien computer that is biological, and there's all these things in the cell, the DNA strand and the mitochondria and all these processes going on, building enzymes, and we have to figure out how this amazing little machine, or a factory, because it makes things all the time, how it works. And so there's such potential there, and it's an exciting for research to figure out how this thing works, and can we control it and do good things with it.
Yes, I think we're stepping right now into a very complicated topic with these ethical issues, especially because things are not only black and white. Usually they have like more gradients of gray in the middle. So for instance, whenever you mention this about changing the color of the eyes of the babies, I just remembered, I live in the Czech Republic, Dr. Tony, and I just remember I saw a couple who were looking for a sperm donator, but the sperm donator, they had a list of characteristics for
Oh
this
boy.
sperm donator,
Yeah.
like tall, blonde, this color of eyes, and like do not have uh, any diseases in the family, running in the family, etc. You know, so that's also something ethical. But on the other hand, if you think about it, for instance, let's say that you discover that your baby has some vision problems, but you have the technology to change that and to give that baby the possibility to have a normal life. So will you do it? Yeah,
Right.
so it's like a very, very complicated issue with this, with the ethical part. I mean,
Yeah, it could improve the quality of life of your child, or it could be used to enforce a kind of racism, you know.
also,
So but I think unfortunately, I think every tool, I mean, the ability to program, a person can make a wonderful new, um, you know, platform for people to communicate like Skype, or they could use their abilities to hack, you know. And so every tool, I guess, could be used for good or bad. So it's but these are really tough issues about what it means to be human and how much have control.
yeah, at the end of the day, I think it depends on all of us how we use it.
Right.
And Dr. Tony, you mentioned that you think that the benefits can outweigh all of the possible bad sides of biocomputing. So could you please like kind of summarize what are the benefits that you see in
Oh yeah.
biocomputing?
Yeah, it's I the benefits that we know of are that envision the cell um, as some people use the metaphor, it's a little factory, okay?
Mhm.
And a benefit could be it can turn a greenhouse gas like methane into oxygen, all right?
Yes.
So there could be benefit with climate. Uh, you could use the cell not to, okay, to create a vaccine, okay,
Oh yeah.
or a
Oh,
medicine.
that's a good one.
And with Dan Gibson's machine, you could send the digital file over there and have them create the actual medicine. So the so that yeah, that's amazing also. So as far as uh, even just being able to read your own genetic code and say, okay, your family, you particular, have a disposition to heart disease, or
Mm.
you could find out in your 20s, so you could start to take better care of yourself, so that when you reach 50 and 60, um, that can, you can help prevent that. So having the knowledge of some people don't want to know. I want to, but some people say, I want to know what I'm prone to. Am I prone to getting dementia when I'm older? And how can I intervene early and stop that? So that's not at the biocomputing level. It's just the ability to read the DNA
To read
sequence.
the DNA. Yes.
Um, and then to write DNA, yeah. Oh my gosh. We could, the way we would do it before, like if you're dog breeding a new breed of dogs, all right,
Mhm.
we would do it the old, like nature would do it. You would take this type of dog and this type of dog, and they would have puppies, and
Yes.
then they would go forward. And I guess you could just um, speed up that process, where it might take 4 or 5 generations of puppies to get this new breed that you wanted, or a new trait in the dog. And now you might be able to do it if you understand all the DNA sequences and what they mean. You might be able to do it in one generation or something,
Yes.
you know. I don't think we can imagine all the benefits right now. But I think those ones of ours, our own health, predicting our future problems, editing, possibly editing our own DNA to help us become less prone to certain diseases. All kind. I'm not sure I can imagine them all. There's so many. It's still so young, really.
And do you know what kind of technologies are used to build these tools? Is it the typical, let's say, C#, Java, or the common things that we know?
Oh, wow.
Or they use something completely different?
Yeah, so on the completely digital side, I don't know the format in which the DNA code is represented. And I'm sure they're using C++, Java, maybe a little Python to actually write the code that manipulates these files, does the algorithms to try to read the DNA code. And you're right. So we're using all the current computing skills and the normal programming languages you have, but then we're also they're connected to, you tell this other machine to put together the DNA strand, and that's a whole other technology. But you're using your computer skills to write the code to instruct it how to do this, these little manipulations. Go ahead.
So do you think we're going to build, like, let's say, our own, how to say it, our own in masses computers that will be, let's say, bio in, I don't know, the next 100 years, replacing what we have, turning into something different?
No, my imagination says that there will always be a hybrid where normal silicon chips and the normal kind of computer, and we type in code, interfaces with some biological thing and instructs it to actually do things and to create this code. So otherwise, well, for one thing, the DNA strand is so small that we can't manipulate it directly. We have to have these layers to get down there, and so I think we'll always be we'll be using C++. I don't know. We'll come up with other languages, computer language, but there'll always be a more electronic or silicon based interface to the biological part. Yes. That's my vision, but I could be wrong. Now, I want to tell you a little bit about some of my research on a different type of algorithm that might be very efficient when it's put into biocomputing. Is that all right to switch?
Sure.
I have other questions now.
No, please go ahead, Doctor.
Okay. So the question I ask, since I'm not a biologist, but I'm learning as much as I can, is basically the cell is a little factory and it can produce things. It can produce things in the world. But normally as computer scientists, we think about, well, can it do computational work, like find the shortest distance between two locations, or the traveling salesman problem, or these more classic ones? Can it help us do that, or are there yet unsolved or unimagined problems it's going to be better at? So that's why So what I've done is try to, I read a number of papers where they try to implement in the cell a logic gate, like an AND gate. Two molecules come down and they're both there, so this and that, and then it releases something, another molecule, okay? Or an OR gate or a NOR gate. So these things.
Yeah, got it.
So that's one way to try to turn the cell into a computer, okay, that we would recognize because it has these logic gates, all right, these circuits. But I'm going off in a very different direction because I think we're arbitrarily constraining the cell and trying to force it to act like an electronic computer or a silicon-based computer. And I say, is there another way that we can leverage, let the cell be more of itself and not constrain it like this, and do computing? So I'm gonna give you an algorithm,
okay,
that is more efficient than the known electronic algorithms. Yeah. And then I'm gonna ask the question, can we get the cell to do this? All right. Okay. So this one is well known. I have others that I haven't published yet, so I'm not going to give you the details of those. But this one is out there, and so I'm going to share it with you. Okay. So suppose you wanted to know the shortest distance between two locations. Okay. And here's a way you could do it. Sounds silly, but here's a way you could do it. You could take your map and you could make put a string model of your map. So between every two locations on the map, you have a string and you tie a knot where the cities are.
Yeah.
Okay. And so at the end, you have this, my hands are gesturing all over the place. You have this string map, a version of your map.
Yes.
So on the real map, if you needed to, it's just a scaled down version of, the real world. Okay. Is that?
Yes.
So
makes sense.
here's what you could do. If you wanted to find the shortest route, let's see, I don't know towns in your countries very well, so I'll stick to the United States. I'm sorry.
Yes.
The shortest route between Boston and Philadelphia, okay? You made a map of that with string and knots. You could just pinch the knot that represented Philadelphia and pinch the knot that represented Boston and pull the string until it becomes tight. And that route, that first string that becomes tight, you could go and say, okay, that's the shortest route between Boston and Philadelphia. And now if I try to trace it, I have to go to this city and get on this highway, and then I have to go to this city, and it is a way to find the shortest route between two locations. Does that make sense to you?
It does.
Now, who would be crazy enough to actually build a string model of a map and go through this? Because we have paper maps and we have all these wonderful algorithms that do it in the electronic way.
Yeah.
That, try out different routes and then eventually come to the shortest one. Okay?
Exactly.
Now, when I do the efficiency analysis, the complexity analysis, the number of steps required to build the map and then to do this pulling thing until it's tight, that becomes more efficient the fewer number of steps, not faster, but the fewer number of steps to perform. It becomes more efficient that way after 10 cities are on the map. Okay?
Yes.
So this algorithm is, some sense, less complex than the electronic ones that search out many variations on the map, because they start from the target and they go out and kind of cascade and gradually get to the destination. So as far as counting the number of steps, this is more efficient. However, we don't have a string computer that can do this quickly, so it's ridiculous to think about it. Okay?
Yes.
However, imagine if I could program a cell to make a network of proteins. Network of proteins that are connected in the way that the map is. Okay? Then I would have to come up with another biological process to take the protein network and pull it, try to extend it or pull it apart, and then retrace the shortest route that becomes tight first.
Okay, yes.
So that's where my research is right now. Can we, and this would work for a lot of network problems. Can we create a protein network that emulates some network we're interested in, and then do measurements upon it, and possibly we could come up with not only it'd be fewer steps, but it could become faster than what the electronic computer is doing when you got to 100 cities or 450 cities or 1,000 cities, right? So that's what I'm looking into. It's pretty wild. It's pretty like very futuristic, but it's very different than what a lot of others, they're trying to force the cell and make logic gates for it to imitate the electronic computer. And I'm saying, no, let's if it naturally makes protein networks, let's try to make networks that are useful to us and extract the information. Because once the protein network is built, the second time you use it, suppose I want to go to another location on the map. I want to go from Charlotte, North Carolina to Chicago.
Okay.
The network, the map is already built, and I don't have to build it up again. I just have to go and find the knot or the location that represents Charlotte and Chicago and pull it apart and then retrace that shortest route. The second time I run this algorithm, it is it's more efficient, or in fact, it's incredibly more efficient than anything an electronic computer can do, because the electronic computer would have to start over. Oh, I'm starting in Charlotte, and I'd have to explore all the possible roads out of Charlotte.
Yes.
And the roads out of those cities, and it would have to repeat its whole algorithm again.
Exactly.
By the protein network, I got the map built. All I have to do is do that second step.
Exactly.
So see? So I'm a crazy man. I'm thinking about these very futuristic things, and I found some. The reason this might be really important is that I have found one of these physical algorithms using string for, excuse me, a problem that's called the subset sum problem. And
Subset sum, yes.
yeah, and that problem is basically given a list of numbers. Can you find a group of them, a subset of them that add up to a particular number that you're interested in?
Oh, I see. Okay.
Now, in
Yeah.
an electronic computer, the best known algorithms are what they call NP complete. There is no efficient algorithm to solve this problem.
Okay.
However, when I do this in my little string model, I have found a string model version that is faster, more efficient, I should say. I got confused. More efficient in the number of steps than the best known electronic algorithm.
Mm-hmm.
So I'm finding these algorithms that if we could get the cell, the protein networks to do this, it would be, we'd find faster and more efficient algorithms for these really hard problems, like the subset sum problem. And there's no efficient solution to it known.
Yeah.
So what do you think of that?
Dr. Tony, I am quite impressed by what we have just spoken. And for me, I have to say that I admire you because you're following a different approach. You're challenging actually what the other scientists out there are doing by, instead of implementing gates, the logical gates, I mean,
Right.
you are right now trying to make use of the already built-in capabilities of the cells in the form of creating the networks of proteins. And it sounds very innovative to me. It sounds very innovative. And I would like to see that we can actually use it for real life applications, such as the ones that you mentioned, the SSP problem.
Yep.
And something that was not that clear to me, and maybe I will ask you to elaborate on that, Dr. Tony. So what is this MP complete problem?
Right. Okay. So yeah, so yeah, and Federico, you can chime in too if you want, because I'm trying to explain it for a more general audience and not get too technical. But basically, when you design an algorithm, like the algorithm that searches from one city and try to find the shortest route to the other,
Yes.
there's you analyze it, and it's called computational complexity. How complex is this algorithm?
Exactly.
And basically, how many steps do you need? And as the network grows, as you add more and more cities, what is the formula that calculates how efficient it becomes? So in the, so let me see. So if it's P, there's two major classes that I'll talk about.
Okay.
P are the efficient algorithms. We found something that is very efficient. Well, it's pretty efficient, okay?
Okay.
And NP stands are the class of that we don't have an efficient algorithm to solve it. It takes a long time. And for example, if the formula is 2 to the nth power.
Yes, I have seen those, yeah.
All right, then 2 to the first is 2. 2 to the second power is 2 times 2 is 4. But then it starts growing very quickly.
Yes.
So 2 to the 100th is huge. 2 times 2 times 2, 100 times. that would be considered an NP problem, because as you increase n, the algorithm takes more exponentially more and more time to solve. And so for huge versions of the problem, if n were 100 or 200, it would take basically, it's impractical to run. It may take hundreds of years, it may take thousands, it may take the length of the universe for been around. So we don't like those algorithms that grow exponentially, and they're in the class called NP. We like algorithms that are in P. So P stands for polynomial, and the simplest, the best kind of polynomial one would be N. If you're dealing with one thing, it requires one step. If you're dealing with two things, it requires two steps. If you're dealing with 10 things, because it requires 10 steps, that would be the best, because as the problem grows in size, the number of steps required grows very in a manageable manner, like in this case is linear. More common, you might be 5n, okay? If you have one thing, 5 times 1 is 5, takes 5 steps. If there's two things, 5 times 2 is 10 steps. If it takes 100 things, 5 times 100 is 500. It's not growing that fast. So the algorithm is, we'll call it efficient, and we like those algorithms. We don't like the other ones. So the one I described to you for the subset sum problem, it's the calculation is 2 to the n, and so that means as the list gets longer, the number of steps required is 2 to the whatever the length of the list is, 2 to the n, 2 times 2 times that many times, and so it becomes very inefficient, and it's put in the NP class. Now the last thing is it's called NP complete, and so there's a group of problems that if you could find an efficient solution algorithm for one of them, you should be able to find an efficient algorithm for all of them. They're all related to each other, this NP complete group, so that solving one of them efficiently would lead to a efficient solution for every one, because they're kind of dealing with the same problem. So underneath. So my little string solution is faster than the electronic algorithms for all those in NP complete. So if I can get this to run in a cell and be very efficient, then I could create other ones that are slight versions of it and solve all these tough problems that don't currently have efficient algorithms for them.
Yeah, and I'm also very impressed by everything that you have told us, Doctor. I didn't have even any idea about all these possibilities. But let's suppose that I am new. How can I get started? Like, how, after listening to you, where should I go? For example, what kind of books do you suggest me, or how to get started?
Yeah, so if you're anything like me, I just had to start out with genetics for dummies to review all the things I learned in 6th and 7th grade, you know, and all the things about it, about what's inside the cell. So you have to start learning about what the cell is doing, and then there are a number of very good TED Talks by Dina Zelinsky about DNA storage. And so they all have a little biology lesson inside these TED Talks. And another one by Dan Gibson, who made this bio printer.
Oh,
They're
nice.
wonderful TED Talks to start saying, well, how can we leverage and use the amazing abilities inside the cell to do good work?
Mhm.
The stuff I'm doing is, yeah, then, so um, the stuff I'm doing is not known yet. I hope.
Mhm.
So, yeah, let's see. What's another one? Another big name. Let me look up on it. I think there's another great textbook
Yes.
about by another author. Gosh, I believe it's I might be wrong. George West. I'll try to figure out if that's correct before we leave. So most people probably interested in this will be computer scientists who have already the programming knowledge and the background knowledge from the computer science side, who want to say, well, what's the potential of this biocomputer? And they'll have to, um, Google biocomputing and then refresh their memory of all the things they learned in biology class and start
Mhm.
looking at it and saying, oh my gosh, it's like a little factory, and maybe we can use it to make great things. And then maybe it's like a little computer, but a very different computer that doesn't use logic gates. Maybe we can use it to solve these tougher problems that involve networks and so forth in a very different way. So yeah, I don't have at the top of my head good suggestions other than to refresh your biology knowledge and build on it from there. The last thing I would say, I'd hope my next phase, after I've created like 3 or 4 of these crazy algorithms, that if we can get it into the working in the cell, it would be very efficient. My next phase is to talk with more synthetic biologists and start moving into who would like to um, try this out. And
Yes.
is it possible to do this with a protein network? And is it possible to do this with these things? So I have to. That's my next move. And if anyone hears this who's in that field, they can contact me because I'd
And
love to chat to try to make this real.
and how you consider to try to reach Elon Musk? You know that he's constantly doing a lot of extreme experiments, and I would be surprised that he could give you the cash immediately or something like that.
Yeah, if he felt it, yeah, that's a great thing with if you can reach these very visionary billionaires, it's nothing for them to write a $1 million check and say, "Oh, go
Mm-hmm.
try that."
Exactly.
That would be fantastic if I could uh,
Maybe,
get some money, grant money, or money from a rich person to see if I can make this real, because the benefits would be really big.
maybe a good idea will be to send him a tweet just to see what is his reaction. I
Okay.
wouldn't be surprised that he might reply. He's constantly thinking in the I don't say even the next big thing, but the next big thing in the next 10 years or even something like that.
Oh yeah, quite a
Very
visionary
visionary,
and
yeah.
quite an active mind. So okay, I will do that. I said, nah, that's not going to work. But Frederico, I'll try it. You convinced me. Thanks.
Amazing. And I would just like to summarize the recommendations you just gave us, Tony, so our
Sure.
audience can listen to them once again. So you mentioned genetics for dummies. You mentioned tech talks about Dina Zelensky
Yeah.
and also Dan Gibson, who is the creator of the bioprinter,
Yeah.
and the textbook. And we might need to double check this from George West. and this is about
Right.
genetics.
I'm looking it up right now to see if, because I have the PDF of his document, and sorry, I wasn't prepared for that. I can't. I mean, I can send it if I find it. I'll send it to you. You can put it up with the podcast if you want, but I can't find it quickly. Sorry.
Okay, no problem. We will add it in the description.
All right, great.
We'll add it in the description later. And now, Dr. Tony, as we usually do here in the Cyber Prophets, that's where we're prophets. We are trying to see how the future will look like. Now, we have already spoken, and this is a totally mind-blowing, very futuristic topic right from the beginning. So I think that we have been speaking about the future most of the time around here. But I would like to still ask you some other questions. So
Okay.
right now we have like our cell phones, our laptops, and everything. Do you think that in the future, biocomputing, or better say like bio devices,
Wow,
will kind of replace what we have nowadays, like bio laptops, biophones, or some kind of devices?
good question. My imagination says that the biological portion of the computer will always be encased by more what we would say normal looking technology.
Okay.
Because we need an interface to this very, very tiny world, right? And
Yes.
even for it to send information to us, we need this interface to go from this very small world up to our size world. And so I always imagined it, how I always imagined it having a regular technology interface. However, I just thought of this, Edgar. I'm going to get really, really wild, futuristic. Okay.
Wow. Yes.
Okay. Ready? I imagined in maybe a sci-fi story, a man who has he's a spy. He has some very secret information, and the police have holding him, and they're searching for the device that holds this information,
Yes. Okay.
and they can't find it because it's in a cell on the back of his hand.
Oh.
Okay. So there might be biological little computers, we'll call them. Imagine they're put in your body. I don't know. Maybe they monitor your bloodstream, and when they detect a certain condition, the organisms in there related release some medication, and maybe there's a little silicon chip with them that communicates, and you send information to that chip, and it interacts with the biocomputer inside your body, and it can do some calculations for you.
Yes.
Maybe, yeah, you could do the shortest distance between two cities, and you would need an interface, but the biocomputer is embedded in your body, or this biocomputer is specific to this algorithm, and this other one is specific to solving this algorithm efficiently. And so you have a whole set of them, and you have a little device that can communicate with the biocomputers that are placed in different parts of your body, and your body keeps them alive and gives them the nourishment they need and the energy they need to. How's that for a wild answer, huh?
That's a very wild answer. Incredible.
Well, it's something that how to say it. I would like to see it. It seems something very interesting that we might be able to see. I don't know, in the next, I don't know, 200 years or something like that.
Yeah.
And in your opinion, do you think that, I don't know, in the next 300 years or 100 years, or when do you see is your timeline that we could have this kind of biocomputer starting as a service? Because I think it will be like the first approach before going to the bio devices.
Yes.
Yes. Oh, well, I'll tell you, the algorithms I have worked out, they could become, yeah. Is that, you made up that term, Frederico? As a service?
Yeah.
Biocomputing as, I like it. I like it. So biocomputing as a service could become a reality in the next, soon, 5-10 years, or if I get a grant quickly from it could be a reality in the next couple of years. Who knows? It's kind of like quantum computing. IBM has made their quantum computer available so people can learn, and you can write a short program and send it and it'll run it.
Yeah.
So that's basically quantum computing as a service. And so why not, Frederico, with your vision? Why can't we have biocomputing as a service? And I think if I get the grant money, it could happen, a year or 2 or 3 after I get the grant money. So I'm going to speculate some form of it will be happening on the next 5 to 10 years. Mm-hmm. And yeah, and that'll happen well before bio devices, but this will be the next step. All right, biocomputing as a service. I love that.
I can see that you're really working hard on this biocomputing topic, Dr. Tony. And now I would like to ask you a question, but this is like more down to earth. Mm-hmm. So what we have now, all the possibilities, all the theory, and what we can do with the CRISPR method for editing DNA. So my question is, like, what is the next step in the development of biocomputing? The right next step.
Right. Well, I just read an article yesterday about another process that can edit DNA, and it seems to be faster and more reliable. And I think it was called introns, INTRONS. Mm-hmm. I need a biochip for my memory. Some of these names I can't remember very well. So I think the next step for biocomputing and synthetic biology is to keep increasing the speed, at which you can read the DNA, write the DNA, and edit the DNA. Mm-hmm. Because right now DNA storage is possible, but I don't think it's going to be commercially viable, only for archiving purposes right now, until you can read and write and edit more faster, more quickly. So I think that's the next step, is to make all these processes that we're doing faster. And then there's so many other things, that little machines or factories going on in the cell, that, I'm exploring protein networks, but, there's other things that I think people are going to say, oh my gosh, the cell is doing that. We could use that. And I don't know what those are. So I think there'll be more practical discoveries of the cell is doing this, and we can leverage it to do something useful for us. not just DNA, not just protein, but other things in the cell. Other processes are going on.
Yeah.
Thank you very much, Dr. Tony. Without a doubt, I hope you will get that grant soon. I hope that your tweet will reach Elon Musk, and if your tweet doesn't reach you, hopefully one of our audiences is going to reach Elon Musk, and we need to become spammers to try to help you accomplish this biocomputing as a service. I really believe that your thoughts, your ideas can empower us to build a better future for everyone. Any final thoughts that you would like to add, Doctor?
Just thank you very much. It's been a delight talking with both of you. And yes, I'm ready to talk to potential grant or people with money and try to make the first device that my theory is articulated. So thank you very much. I enjoyed this.
Thank you.
Thank you very much for everyone, and hopefully we'll see you soon, where we keep prophesizing more technologies and you can develop a better future with everything you're learning. See you the next time.